Good morning, everyone, and welcome to the Primary and Acquired Resistance to Cancer Immunotherapy session at the Immuno-Oncology event. I'm Leo Nissola, a medical doctor and scientist at the Parker Institute for Cancer Immunotherapy, and I'm a former MD Anderson Fellow. My work today focuses on clinical development, accelerating research, and exploring novel approaches to tackle cancer with immunotherapy at PICE. One of the main challenges in cancer immunotherapy is the resistance to IO therapies. Primary resistance can occur before treatment, and acquired resistance can occur in patients who responded to treatment but relapsed after a period of effectiveness. Most cancer patients will develop primary resistance to checkpoint inhibitors, while 40% of those who don't have primary resistance have an objective response. Although immunotherapy can induce tumor cell death and provoke significant clinical results, there are still unknown mechanisms that limit the application of cancer immunotherapy. I have no actual or potential conflict of interest in relationship to this program or presentation, and I'm speaking on behalf of the Parker Institute of Cancer Immunotherapy. Some high-level topics that we'll be covering in this talk today are related to the progress that we've made in immunotherapy. What are the different types of resistance and the strategies in which we are approaching immunoresistance? Through the years, we have made a lot of advances in immunotherapy, and focusing specifically on immune checkpoint therapy. Monoclonal antibodies targeting co-stimulatory immune checkpoints, such as PD1 and CTLA4, have demonstrated clinical activity in several malignancies, including melanoma, non-small cell lung cancer, renal cell carcinoma, bladder cancer, head and neck, squamous cell carcinoma, MSI-high colorectal carcinoma, Merkel cell carcinoma, and so on. Clearly, immunotherapy has changed the practice of medical oncology. And I think in the past year, we saw an approval in breast cancer for the first time, which is very exciting, especially because for many people, we are treating breast cancer previously. And in prior trials, clinical efficacy in immunotherapy did not go so well. So it was a very happy moment to see that happening last year for breast cancer patients. Checkpoint inhibitors have transformed cancer treatment, but many patients still do not respond to it. Human-derived multi-omics studies have shown immunological distinct class of resistance to PD1 therapy, and future work should focus on understanding immune surveillance at a single-cell level. Patients who have primary resistance to checkpoint inhibitors do not respond to initial therapy. Ongoing studies indicate that both tumor cell intrinsic and tumor cell extrinsic factors contribute to resistance mechanism. And the most straightforward reason why a tumor would not respond to immune checkpoint therapy is the lack of recognition by T cells because of the absence of tumor antigens. Alternatively, cancer cells may have tumor antigens but develop mechanisms to avoid presenting them on the surface restricted by MHC, due to the alterations in the antigen-presenting machinery. Early trials with monoclonal antibodies targeting PD1 and PD-L1 have demonstrated durable clinical responses in patients with non-small lung cancer. However, current assays for the prognostic and predictive role of PD-L1 expression are not standardized with respect to either quantity or distribution of expression. Objective determination of PD-L1 proteins reviews heterogeneity within tumors and prominent interassay variability or discordance. This could be due to different antibody affinities, limiting specificity or distinct target epitopes. Efforts to determine the clinical value of those observations are underway, but we know that PD-1 expression is heterogeneous. PD-L1 is mainly expressed on the surface of tumor cells and antigen-presenting cells in various malignancies, such as squamous cell carcinoma, carcinomas of the brain, GI tract, bladder, ovary, and the skin. In the tumor microenvironment, PD-L1 expression on tumor cells and other tumor-promoting cells is caused by two mechanisms: constitutive mechanism and induced mechanism, both of which depend on the binding sites of IRF1. For example, BRAF-mutated melanoma, the PD-L1 expression is a result of cancer cells' adaptive response to immune attack evoked by cytokines, or a constitutive expression which is a result of oncogenic processes. PD-L1 is rarely expressed on normal tissues, but inducibly expressed on tumor site, which makes PD-L1 pathway uniquely different from any co-inhibitory pathways, indicating that the selective expression of PD-L1 may have some association with clinical outcomes for cancer patients and can be a selective target for anti-tumor therapy. So, dissecting the tumor microenvironment during disease regression and progression could show us a little bit more of that understanding. As you can see in this picture, A, expression scores of genes related to tumor microenvironment, immune cell types, or even immune cell states. The blue backgrounds indicate biopsies taken during disease regression, and orange backgrounds indicate biopsies taken during disease progression. On picture B, samples for each time point from tumor were stained with specific antibodies against CD8 or CD4, and were quantified using a cell counter function. Boxes in the upper panels are shown at a higher magnification in the lower panels. Advancing treatment options for patients with primary resistance to immunotherapy requires a pursuit of understanding of what are the biomarker subtypes of resistance to PD-1 therapy, or how are these biomarkers different across cancer types and treatment regimens. And we need to have a clearer understanding if the body of knowledge that we have developed would allow us to understand the related toxicities and the association with resistance to PD-1 therapy. Identifying the potential relationship between the tumor microenvironment, the tumor mutational landscape, and the PD-1 resistance could help us develop such biomarkers and perhaps create a predictive model for toxicity and IRAEs. Here, some basic science that led us to where we are in terms of immune checkpoint blockade. And as many of you are no doubt aware, engaging a T cell response is a multi-step process. And it really involves, at the very least, the interaction of a T cell with an antigen-presenting cell. After the T cell becomes activated, CTLA-4, for example, translocates from this intracellular store into the immune synapse, where it binds, in fact, with 20-fold higher avidity to the same co-stimulatory molecules that CD8 28 does. And instead of generating a positive signal like CD8 28, it generates a negative signal. And so, with that knowledge in hand, Jim Allison theorized that if you would block CTLA-4, such as with an antibody, you would allow a T cell to become more proliferative and more activated than normal. This sort of physiological governance allowed and really led the hypothesis of antibody-based immune checkpoint blockade to the first agent of its class. Immune checkpoint inhibitor therapy has been particularly successful in melanoma, for which approved therapies include PD1, anti-PD1, anti-CTLA-4, and the combination of both regimens. Long-term survival data for patients with melanoma treated with ipilimumab indicates 20% of patients show evidence of continued durable disease control or response after 5 to 10 years after starting therapy. The response rate for melanoma patients treated with pembrolizumab was 33% at 3 years, with 70% of patients initially responding, maintaining a clinical response, as we saw in a paper from Tony Ribas in 2016. Combination therapy or dual immune checkpoint blockade has recently shown dramatic responses in patients with metastatic melanoma with RR of 58%. However, half of patients experience significant toxicity from the treatment regimen, and survival benefit for this approach has to be demonstrated. The incidence of metastatic melanoma has increased over the past 3 decades. Regulatory pathways that limit the immune response to cancer are becoming increasingly well characterized. Cytotoxic T lymphocyte-associated antigen 4 is an immune checkpoint molecule that downregulates pathways to T cell activation. This fully human monoclonal antibody that blocks CTLA-4 promotes anti-tumor immunity and has shown activity in patients with multi-tumor types, including melanoma. Challenges remain in defining what responders and non-responders are, especially given the heterogeneity in patterns of response with checkpoint inhibitors. Such heterogeneity can be spatial, meaning a different response in different lesions, or temporal, or stable disease followed by progression, manifesting within a given patient mixed responses. Oligometastatic progression and or stable disease with isolated progression can be seen as well. And despite these complexities, the tail on the survival curve suggests that long-term disease control is possible in a significant proportion of patients successfully treated with checkpoint inhibitors. This possibility of expanding this long-term clinical benefit to more patients with advanced cancer is thus fueling more and more investigation into the elusive mechanisms of response and resistance to this therapy. Patients who have primary resistance to checkpoint inhibitors do not respond to initial therapy, and ongoing studies indicate that both tumor-centric, cell-centric, and tumor cell extrinsic factors contribute to these resistance mechanisms. The most straightforward reason why a tumor would not respond to immune checkpoint therapy is the lack of recognition by T cells because of the absence of tumor antigens. Alternatively, cancer cells may have tumor antigens, but develop mechanisms to avoid presenting them on the surface restricted by MHC. The cell intrinsic factors that contribute to immunotherapy resistance include expression or repression of certain genes and pathways in tumor cells that prevent immune cell infiltration or function within the tumor microenvironment. These mechanisms may exist at the initial time of presentation, which highlight primary resistance mechanisms, or these mechanisms may evolve later, which will be highlighted by the adaptive resistance mechanisms. Multiple tumor intrinsic mechanisms have recently been identified, and they include signaling through the mitogen activated protein kinase pathway, or the loss of PTEN expression, which enhances PI3K signaling, or the expression of WNT beta-catenin signaling pathway, or the loss of interferon gamma signaling pathway, or the lack of the T cell response due to the loss of the tumor antigen expression. Oncogenic signaling through this pathway results in the production of VEGF and IL-8, among many other secreted proteins, which have known inhibitory effects on the T cell recruitment and function. Similarly, the loss of PTEN, which enhances PI3K signaling and is the most common phenomenon across several cancers, including 30% of melanomas, was found to be associated with the resistance to immune checkpoint therapy. Importantly, the frequency of PTEN deletions and the mutations was higher in non-T cell inflamed tumors as compared to T cell inflamed tumors. In addition, murine models lacking beta-catenin responded effectively to immune checkpoint therapy, whilst beta-catenin positive tumors did not. Non-T cell inflamed human melanoma tumors, which lack T cells and CD103, dendritic cells in the tumor microenvironment had significantly expressed higher expression of tumor beta-catenin signaling genes. Cancer cells that constitutively express immune suppressive cell surfaces ligands like PD-L1 may actively inhibit anti-tumor T cell responses. Tumor cell extrinsic mechanisms that lead to primary and adaptive resistance involves components other than tumor cells within the tumor microenvironment, including regulatory Tregs, myeloid-derived suppressor cells, macrophages, and other inhibitory immune checkpoints, which may all contribute to the inhibition of anti-tumor immune responses. Regulatory T cells, which can be identified by expression of FOXP3 transcription factor, have a central role in maintaining self-tolerance. The existence of suppressor T cells could downregulate immune responses of an antigen-specific T cells, and it was first identified nearly 4 decades ago. Published data indicate that many human tumors are infiltrated by Tregs. And I wanted to highlight some of the PICE projects that are trying to tackle this understanding of immune resistance. First is exploratory study with nivolumab with or without ipilimumab, according to the percentage of tumor CD8 cells in patients with advanced metastatic cancer. In this prospective study, patients will first undergo tumor biopsies, and patients with a higher concentration of T cells will receive a single checkpoint inhibitor, nivolumab. Patients with a lower concentration of T cells, given a cold tumor, will undergo a combination of nivolumab plus ipilimumab. Through a battery of tests, which include gene sequencing, protein analysis, imaging, and gut microbiome analysis, we will evaluate how the immune system is changing before, during, and after treatment. This uniquely comprehensive collection of biomarker data will allow us to create a heat map of cancers and of the cancer, and it could guide us further studies and potential treatments in the future. Here, this is a unique collaboration between the Parker Institute, BMS, CRI, and biotech partner Apexigen. It's a phase 1b/2 study evaluating the safety and efficacy of CD4 antibody, nivolumab, and chemotherapy. The study first opened in July 2017, and this immunotherapy combination has shown preliminary results demonstrating encouraging clinical activity across cohorts in first-line metastatic pancreatic adenocarcinoma. Here we are seeing a prostate cancer platform study, and as you know, prostate cancer is an immune desert. So given this tumor is cold, we are approaching different multiple drug immunotherapy combinations. First, activating the immune system with pegylated IL-2 by enhancing the number of tumor-infiltrating lymphocytes, and perhaps overcoming adaptive immune resistance with the backbone of anti-PD1. Another approach is inducing immunogenic cell death with radiotherapy by mobilizing and activating dendritic cells with a FLT3 ligand, and stimulating the immune system with a PMP adjuvant. And to the right, we see a DNA-based vaccine with 3 plasmids delivered by electroporation, IL-12, PSA, and PSMA. And the overarching idea here is to mobilize and activate dendritic cells, drive killer CD8 T cells, and perhaps turn this cold tumor into a hot tumor with immune infiltrate. Last year, on March 8th, the FDA granted an accelerated approval to atezolizumab in combination with paclitaxel protein for adult patients with unresectable locally advanced or metastatic triple-negative breast cancer whose tumors express PD-L1. In patients whose tumors tested positive for the expression of PD-L1, the median overall survival at atezolizumab was 25 months, compared with 18 months for those who received nab-paclitaxel and placebo. This approval for atezolizumab used in combination with chemotherapy is a great win for breast cancer research and an extraordinary win for immunotherapy. However, treatment response for cancers to immunotherapy is very difficult to predict. Positron emission tomography imaging may help to predict this treatment response. PET will evaluate immunotherapy targets and markers of immune activation, has shown some promises in early clinical trials. Antibodies and small molecules have used PET for different imaging characteristics, and clearly more studies are needed to better interpret and validate PET scans for this purpose. Biotech companies such as ImaginAb are developing a pipeline of imaging products that leverage the exquisite specificity of monoclonal antibodies and create precise images of disease activity. So scientists have re-engineered antibodies into fragments called minibodies. And those are linked to a radioisotope to enable a safe and rapid, detailed illumination of the targeted cells. This ensures that the minibody is inert and it doesn't exert any additional effects on the body aside from illuminating these relevant targets. So patients on these clinical trials have been receiving imaging agents prior to a routine full body scan with PET. And this technology would produce images that provide a full body view of the disease activity and the drug response, which could offer important insights at critical points of the patient journey. This particular investigation, CD8 positron emission tomography tracer zirconium 89ZR, which is in clinical studies, have demonstrated to have an acceptable safety profile and to be a sensitive PET agent that has the potential to detect the presence of CD8 cells in tumor and T cell rich tissues as a tumor agnostic biomarker. Biomarkers are very important, especially because they help us guide and select patients according to their tumors and what the treatments may work best for them. And I think this all came about in an era of genomics, genomic medicine, where we were really basing our treatment strategies on specific mutations within a patient's tumor. And it has been something that we've been struggling a little bit in the immunotherapy world because the immune response is so dynamic. Part of developing therapeutics is the need to identify patients who will respond to treatment. Thus, to advance drug development in precision medicine, there is a need to develop technologies that are more objective and quantitative through the collection and integration of larger data sets. Multiplex ion beam imaging is a novel imaging technology. And these methods characterize the prevalence, location, and expression of relevant biomarkers that are critical to the successes of targeted therapies. There's also some anecdotal evidence for all PD1, anti-PD1, and anti-PD-L1 therapeutics that patients negative for these biomarkers by IHC respond to treatment, indicating, again, that multiple markers and quantitation will be needed to capture ideal responders. However, chromogenic staining is fraught with serious drawbacks that influence scoring, which is why, additionally, we need to expand those investigations into the integration of larger data sets, and MIBI works well for that. T cell response to checkpoint blockade relies on the expansion of a distinct T cell repertoire that may have only recently entered the tumor. However, whether the T cell response to checkpoint blockade relies on reinvigoration of pre-existing tumor-infiltrating lymphocytes or on the recruitment of novel T cells remains unclear. Tracking a T cell receptor clones and transcriptional phenotypes revealed coupling with tumor recognition, clonal expansion, and T cell dysfunction marked by clonal expansion of CD8 T cells, which co-express markers on the chronic T cell activation and exhaustion. There are significant efforts underway to identify reliably predictive biomarkers of response and resistance to checkpoint inhibitors in baseline tumor biopsies in patients on immune checkpoint blockade. To date, the best predictive biomarkers identified include total tumor mutational load, as well as the markers on an effective immune infiltrate within a tumor, signifying a hot tumor microenvironment, typified by the increased number of cytotoxic T cells, lymphocytes, in proximity to the PD1 positive cells. Mutational load is highly relevant, as tumors with a higher mutational load exhibit higher levels of neoantigens capable of inducing anti-tumor immune responses and translating into a higher likelihood of response to immune checkpoint blockade across several cancer types. Integrative approaches incorporating analysis of several of those features have also been developed, such as a cancer immunogram, which incorporates analysis of 7 distinct features within a tumor microenvironment: tumor sensitivity to immune effectors, tumor foreignness, general immune status, immune cell activation, infiltration, absence of checkpoint molecule expression, absence of soluble inhibitors such as interleukin-1 or 6, or the absence of inhibitory tumor metabolisms. An emerging strategies in elucidating mechanisms of response and resistance to immune checkpoint blockade involves the assessment of longitudinal tumor samples throughout the course of treatment. So here I end my talk. Thank you for watching our discussion. Please feel free to send my way questions or comments. And I would just like to give a shout out to our clinical development super team at the Parker Institute. And even in challenging times, such as those that we're living now, we are working at full steam to speed up science and deliver clinical trials to academic centers across the country. While we are working remotely, we continue connected, pushing science forward, one clinical study at a time. And a great thank you to all of our academic centers that are in the trenches with patients taking on the toughest cancer types. As we usually say back at Pisces Central, cancer fears spicy. Thank you.